Good morning, everyone. Uh, my name is R.Mustafa. I'm working in Molecule CRO in Jordan and going to talk today about PE studies in Jordan. Actually, I'm glad to be at the Biopharm Scientific Annual Meeting, and hopefully we can be in person for the coming years. To start with, this is the outline of my presentation. We'll talk about some terminology about bioequivalence, a little bit of history, and we'll talk about the regulatory aspects of PE studies in Jordan, along with issues and deficiencies that the committee of registration, the most common actually issues and deficiencies that the committee of registration and the Jordanian FDA have encountered during the review of studies that have been submitted to them. Of course, we'll talk to start with about the bioavailability and the bioequivalence. Bioavailability actually is the rate and extent to which an active ingredient or active moiety is absorbed from a drug product and becomes available at the site of action. And the bioequivalence is the absence of significant difference in this bioavailability in two formulations, two pharmaceutical equivalents or alternatives under similar conditions in an appropriately designed study. The pharmaceutical equivalents contain the same amount of the same active substance in the same dosage form that meet the same or comparable standards. And of course, we know pharmaceutical equivalence doesn't mean bioequivalence in necessity. Pharmaceutical alternatives, these are the medicinal products with different salts, esters, ethers, isomers, mixtures of isomers, complexes, or derivatives of an active moiety, or which differ in dosage form or strength. So when we talk about bioequivalence, the significant component of therapeutic equivalence is a bioequivalence study along with a pharmaceutical equivalence. So we said that pharmaceutical equivalence doesn't mean bioequivalence. We need bioequivalence along with the pharmaceutical equivalence to talk about therapeutic equivalence between two drugs. The reference drug that is used and the generic drug, of course. To achieve bioequivalence, we need an in vivo dissolution model. We compare the products and we have a comparison of the product characteristics to ensure therapeutic equivalence. What forms of PE studies are considered in Jordan? Or let's say internationally. Actually, Jordan is working according to the international framework in the world. And of course, harmonization of the guidelines along the world is a requirement right now. So we have the in vivo PE studies, the pharmacodynamic studies, some sort of clinical studies, or in vitro methods. That would depend on the characteristics of the active substance and the dosage forms. We might just illustrate this later on. Let's talk about the history. I'm working in this field since 1999. And actually, in 1985, uh, there were no Jordanian FDA at that time, but there was the Drug Directorate under the Ministry of Health in Jordan, and it required PE studies to be submitted only for narrow therapeutic index drugs. Starting 2000, and especially with the huge number of pharmaceutical companies that started working in Jordan, and with the presence of BE centers, or let's say CROs that are conducting bioequivalence centers, bioequivalence studies, in Jordan, there was a need to have the PE studies required for generic drugs and to issue laws that regulate these PE studies on both sides. The, um, the clinical studies law that were drafted released as draft at 2001, and as well the requirements of PE studies to be registered and for the medication to be registered in Jordan as well. It was drafted in 2001. The applicable guideline for this requirement now was released in 2017. What about the regulatory aspects in Jordan for PE studies? We have, as I told you before, that both laws, criteria, and standards related to the evaluation of BA/BE studies for the year 2017 and the clinical studies law number 2 for the year 2011. Let's talk about it as a story first. What's happening is having the CRO or the Bioequivalence Center in the same place with the pharma companies helped a lot in creating that story. Study drug approval by the Jordanian FDA or the drug directorate at that time, and then the drug directorate was transferred under the Jordanian FDA in 2003 when it was first initiated. And the other side is, what do we have rules and guidelines and requirements for submitting PE studies in Jordan? Especially that if you talk about the medications in Jordan and the pharmaceutical industry, they one of the 10 top experts in Jordan is medications. It's about 8% of what Jordan exports to our other countries. So we have first to talk about is the format and contents of bioequivalence reports that are being submitted. This is a checklist that is available in Appendix 1 under the guideline. We can take a look here. The requirements of comparative dissolution for waiver requests is also present under this guideline. Format and content for comparative dissolution is also present. Clinical study report acceptable, other cases than PE also present under this guideline. Bioequivalence studies on generic products are usually conducted on the highest approved strength, unless there were safety concerns regarding using the highest approved strength. The pharmacokinetics showed nonlinearity between the different strengths, and the reference of the highest strength was not registered internationally. Because one of the requirements that the choice of this reference is appropriate is being registered internationally. When we don't require PE studies to be conducted, of course, when we have an aqueous solution for parenteral use containing some active substance in the same concentration and excipients that do not affect the kinetics within the biological system. And in case of products in aqueous solution and contain active substances in the same concentration as an oral solution currently approved, and the excipients in the product wouldn't affect the GI transit absorption of drug substance. Gas for inhalation, ophthalmic products prepared as aqueous solutions with topical effects, powders for reconstitution as a solution containing the active substance in the same concentration. Of course, these are not optimal because sometimes there are some effects, systematic effects, which would require at this specific time to submit a study. We'll talk about it later on. Studies to be submitted in Jordan. Bioequivalence studies for immediate release formulation with systemic action. All studies performed with test product of same composition and manufacturing process of the reference product. Of course, you should have full reports pivotal for fasting and fed states, bioequivalence or comparative bioavailability studies from formulation development. The study should be conducted on biobatches. The pilot batches may be used provided that they are not smaller than 1 over 10 of the expected full production batch. The same manufacturer, same composition for product intended to be marketed. If different, the post-approval changes or technology transfer guidance should be applied. For the immediate release forms of any medication, a single dose BE study in fasting state is adequate. In case that food enhances or interferes with the drug absorption, a bioequivalence study in fed state should be performed. That, of course, refers to the SMPC of the reference drug. If it states that take it with or after food, let's say, you should go to the fed state study immediately. Modified release drugs, including pellets and beads, of course, we should have both fasting and fed condition to ensure absence of dose dumping. If we have an innovator that is developing a product with immediate release formulation to a generic with modified release formulation, okay, they should submit BE study in fasting state for all the concentration with fed and steady state for the higher concentrations only. Lower strength BE requirements can be waived, which means that we can waive asking for a lower strength BE study, and for us, the higher strength is okay. For immediate and modified drug release drug products, in vivo bioequivalence is demonstrated on the highest strength. Qualitative composition of the different strengths is the same, and the products are manufactured within the same process and manufacturer. The composition of the strengths are quantitatively proportional, and of course, this will lead us to linear PK profile over the therapeutic dose range. In addition to the in vitro dissolution profiles of the test products are similar. We have biowaivers. When in the immediate release forms, of course, we have, according to the BCS, class 1 drug substance, provided that it's not narrow therapeutic index, of course, with a linear PK. And class 3, drug substance that's highly soluble and low permeable. And of course, there are some eligibility criteria that will be evaluated by the committee when submitted. Comparative in vitro. For BE, comparative dissolution between test and reference in compendial media or applicable media for batches used in the BE study, and for the waiver request to lower strength, for the immediate release, the data should demonstrate similarity of the solution profile between the lower strength and the higher strength of the test product, exception for the low soluble with reference products. Modified release, data should demonstrate the similarity of the solution profile between the lower and the higher strength of the test and with same concentration of the reference drug as the concentration of the test drug. For locally applied products like nasal, ocular, dermal, rectal, vaginal, etc., without systemic absorption, pharmacodynamic or clinical studies are required. If the product has systemic effects, of course, a PE study is required. As well, when we do require other studies other than bioequivalence, as well and the replacement therapy for endogenous substances in the body, intranasal formulations, or some inhaled preparations, inhalers. What is the study approval process in Jordan? let me tell you this. The Jordanian FDA has its own guidelines to accredit CROs that are conducting bioequivalence studies. These accreditation would be for the clinical site that the study is conducted in, the bioanalytical laboratories that the biosamples are being analyzed in, and the diagnostic labs or clinical labs where the tests for the study subjects are being conducted, and for the IRBs as well. So if you go to the Jordanian FDA website, you can find a list of accredited clinical sites, centers, and IRBs, labs, and you can use any of them to conduct your study, whether, of course, whether it's a PE or a BD or a clinical study or whatever type of studies you are going to conduct. The BE study should be submitted to the Jordanian FDA by these accredited centers before the conducting of the study and after getting the IRB approval. Usually it takes 1 to 2 weeks after submission, maximum, of course. If there are any comments and any need for amendments, so you can go back to that committee. It would take like 2 weeks. This applies only for PE studies, but for clinical studies, it takes a longer time. As the clinical studies committee, who's responsible for reviewing the protocols, are reviewing the protocols before the study conduct, we have our own committee as well that reviews the BE study report after the conduct of the study. Of course, we are talking about when we are talking about the registration committee, the committee who reviews the reports, it's totally for every company that is applying to register their medication in Jordan. So they do the evaluation based on some criteria, like what's the name of the product? Is it an aerotherapeutic index or not? Is there any variations? Because it's been dealt with like case by case. What's the dosage form? Is it immediate release, modified release? We check the certificate of analysis, formula number of both drugs, batch number, expiry date, type of batch, is it pilot, scale, commercial, confirmation that the formula has no changes, we don't have any variations, we don't need any technology transfer in here, comparative dissolution profile for 3 batches, and we look at the study design, protocol, and date of the study. Was the protocol designed properly? Subjects, how did they select the subjects? Are they healthy? Are they patients? What that was based on what? The selection of the subject, inclusion, exclusion criteria, the number, the sample size that we are using in the study, how many subjects? Are there withdrawals? How many? Why did they withdraw? Was there any adverse events or safety concerns that must be taken into consideration? The outliers, of course. If we have any outliers in the results, they look at the outliers. Are they included in the PK and statistics or not, and why? The study conduct. Starting, was it fast or fed? Does it go according to the guidelines? Is there any proper justification if not? This is very important because there is nothing actually is specified in our guideline. It all depends on the scientific and regulatory evaluation of the committee. And of course, with the justification and the references that has been applied by the marketing authorization holder. Study parameters BK, are they only AUCs and Cmax? Is Tmax included? For some drugs, yes, it is. As well, was the study properly designed actually? Did it has any specific guideline for conduct or even for PK parameters? You know, we can find many of product-specific guidelines than EMA and US FDA. Is the reference product suitable? Was the study designed such that variability due to factors other than the product was reduced? Other design issues like sample size, sampling protocol, adequate bioanalytical method validation. Is it um, is it applicable as was it conducted? Sorry, as per applicable regulations? Appropriate PK analysis, what software it was done on, the statistician did a proper interpretation. After this evaluation is being done by the committee, there are some issues and deficiencies, of course, that they might get out with. These deficiencies will be sent back to the marketing authorization holder, and the marketing authorization holder, if needs to cover some deficiencies in the study conduct by the CRO, they go back to the CRO where they conducted the study. For example, justification of FED study. Um, some sponsors might apply only a fasting study, a fasting state study without a FED study. However, it says somewhere in the SMPC that it might affect the PK of the drug in the body, or take it with food, and it was not taken into consideration. The certificate of analysis, the assay difference between the test and reference, was it less than 5% or it was more than 5%, higher than 5%, and what's the justification provided? Are there any applicable references for that? The outlier results not included in statistics. Stability, long-term stability, especially we are talking about long-term stability. That some CROs might conduct the clinical part, and then the validation would have the long-term stability like 3 months, and it might be should be starting day one from having the samples on in the clinical site till the last sample that has been analyzed more than 3 months. A long-term stability section should be added to the validation report, and of course, it should be successfully calculated. It should shows that this method shows the stability of the drug and the plasma or on the biological matrix we're using till that time. Has the study been conducted in accredited qualified BE centers? This is very important for Jordanian and FDA. They ask for GCP certificates, GLP certificates, maybe because it's in Jordan. What we do is we have GCP and GLP certificates for our CROs or PE centers. In the case you don't have any, but at least we should have some inspection reports that shows the qualification of the center you are conducting your biostudy in. Was the protocol properly designed? The sample size is enough. You're not applying to register your medication with a pilot study, for example. The IRP approvals, are they obtained? Are they originally obtained, and are they obtained before the study conduct? This is very important, of course. The Cmax range widening is something common that some people would do it. And of course, this would not be acceptable to Jordanian FDA without a scientific justification. Biobatch versus the proposed production batches. Are they of the same manufacturing process? And the bioanalytical part, has the study showed or demonstrated bioequivalence based on the parent compound or one of the metabolites? You can refer to guidance, specific product specific guidance on that, and you should have your own scientific justification to apply, because it's well known that the bioequivalence can be demonstrated mainly by depending on the parent compound bioanalysis. Here we have just finished our story today, which started, let's say, in 2000, along with pharma companies in Jordan and drug directorate under Ministry of Health in 2000 and under Jordanian FDA in 2003, and with pharma companies, pharma industry in Jordan. So overall, we have like 15 uh, I think 25, 15 to 25 pharma industry, pharmaceutical manufacturing companies. Some of them are well known. And we have five centers to conduct main 5 centers to conduct bioequivalence studies, and many actually are registered as for clinical studies. It was a pleasure being with you in this little time, and hope we'll see you again next year in person. Thank you so much.